Welcome back to Dr. Liu's VCE Biology. Today's topic is cellular biology, as presented in the 2019 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The cytoplasmic membrane is a hydrophobic barrier made of a phospholipid bilayer. This structure is selectively permeable, allowing small, hydrophobic molecules, such as oxygen, to pass through, but blocking larger, hydrophilic molecules, including sodium ions, polar proteins, and ribonucleic acid. A signaling molecule, also known as a hormone, is produced by a source cell and travels to a distant target cell within the same body. An antibody is not a signaling molecule because it is specifically designed to bind to an antigen. A pheromone is secreted outside the body to act on other organisms, and a neurotransmitter acts specifically on the nervous system and does not travel to distant target cells. Because this signaling molecule passes through the cytoplasmic membrane, it is hydrophobic. It then binds to an intracellular receptor, and the resulting complex travels to the nucleus. There, it triggers the transcription of a target gene, which is then translated into proteins. This mechanism leads to increased gene expression, but does not involve signal amplification or the production of second messengers. The scanning electron micrograph below shows a cell in the early stages of programmed cell death. The cell is beginning to change shape, and blebs can be seen on the plasma membrane. Signals originating from within the cell or from its external environment can both trigger programmed cell death. Internally, three key signals initiate this process: damage to mitochondria leading to the release of cytochrome C, the activation or release of caspases, and the destruction of crucial organelles like the plasma membrane. To maintain a healthy multicellular organism, programmed cell death is essential. This process serves to eliminate damaged or non-functioning cells and to dispose of cells that are no longer required, such as the plasma cells in the immune system, or the cells that form the webbing between the digits. While the removal of infected cells is a valid function, it should be noted that this process is specific to virally infected cells, not other pathogens. Excessive programmed cell death, apoptosis, is caused by an imbalance in the cellular signaling pathways that regulate it. This can happen due to overproduction of death signals. The body creates too many signals that tell cells to die. This includes an increase in trigger molecules like TNF2 or a higher number of death receptors on the cell surface. Reduced survival signals. The body produces fewer molecules that stop cell death. A decrease in inhibitors like BCL2 means there's less protection against the death signals, causing cell death to proceed unchecked. Overactive executioners, the enzymes that carry out the cell's destruction, called caspases, become overactive or are produced in excess, leading to rapid and widespread cell death. Genetic changes, mutations in genes can cause an imbalance in these signaling pathways, for example, by producing too many death receptors or non-functional inhibitors. This genetic malfunction leads to excessive cell death. The question required students to explain what brought about excessive cell death. However, many students explained why cell death occurred. Botulinum toxin is produced by the bacterium Clostridium botulinum. In large concentrations, botulinum toxin causes the disease botulism in humans. Botulinum toxin gains entry into nerve cells through endocytosis. The correct diagram should have depicted endocytosis, A process where a section of the plasma membrane folds inward to engulf the material to be taken up, in this case, the toxin, forming a vesicle inside the cell. Students were expected to label both the membrane and the toxin. A diagram incorrectly showing the vesicle outside the cell was a common mistake that only earned 1 mark. A number of students also mistakenly illustrated exocytosis, a process opposite to what the question asked for. Upon entering a nerve cell, botulinum toxin binds to and affects a specific protein, which in turn inhibits the release of neurotransmitters. The target protein has a complex quaternary structure made up of 4 distinct polypeptide chains. The toxin disrupts this structure, causing the polypeptides to detach from each other, effectively denaturing the protein and rendering it non-functional. Botox injections contain weakened botulinum toxin that the immune system recognizes as a foreign antigen. The body responds by producing specific antibodies and creating memory B cells. With subsequent injections, these memory cells trigger a faster and stronger immune response, leading to the rapid neutralization of the toxin. This quicker immune reaction is why the muscle paralysis from later injections lasts for a shorter period. This section is worth a total of 15 marks and accounts for 12.5% of the overall exam grade, 120 marks total. The content focuses on transport across the plasma membrane, apoptosis, endocytosis, and the denaturing of proteins. Multiple choice questions, 3 marks. Question 1, 1 mark assesses the understanding that hydrophobic molecules are the only type of molecules that can freely diffuse across the plasma membrane. Question 18 and 19, 2 marks focuses on hormones as signaling molecules. Specifically, it tests the concept that hydrophobic hormones can diffuse across the membrane and trigger the expression of target genes. Short answer questions, 12 marks. Question 2, 6 marks. This question has 3 parts related to apoptosis, programmed cell death. A asks about the internal signaling molecules that initiate apoptosis. B explores the role of apoptosis in maintaining a healthy organism. C requires an explanation of how the malfunction of apoptosis can lead to disease. Question 4, 6 marks. This question focuses on Botox and has 2 parts. A examines how Botox enters a cell via endocytosis and then denatures its target protein. B tests the understanding of how the immune system responds to Botox by producing memory B cells, which leads to a quicker clearance of the toxin upon subsequent injections. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.